The objective of this experiment is to determine the content percent of phosphorus in plant food through gravimetric analysis. The plant food sample was obtained, and the set of numbers written on the label was recorded. The plant food sample was weighed between 3.0 to 3.5 grams using a weighing paper. The sample was transferred to a 250 milliliter beaker. Between 35 to 40 milliliters of distilled water was added, and the mixture was stirred with a glass rod to dissolve the sample. The insoluble material was removed through filtration. To the filtrate, about 40 milliliters of 10% magnesium sulfate heptahydrate was added. Then about 100 milliliters of 2 molar ammonia aqueous was added slowly while stirring. The mixture was allowed to sit at room temperature for 10 minutes to complete the precipitation. A filter paper was accurately weighed. The paper was folded and fitted into a glass funnel. It was wetted with distilled water to hold it in place. The precipitate and the entire solution were carefully transferred from the beaker onto the filter paper. The precipitate was washed by adding 3 portions of 5 milliliters distilled water to the beaker. It was then rinsed with 2 10-milliliter portions of 75% isopropyl alcohol through the filter paper. The filter paper was removed and placed on a numbered porcelain evaporating dish. It was dried in an oven at 60 degrees Celsius for 24 hours. Once the magnesium ammonium phosphate hexahydrate was thoroughly dry, the filter paper along with the precipitate was weighed. The mass was recorded, and the percentage of phosphorus in the original sample was calculated. This experiment aimed to determine the percentage of phosphorus in a plant food sample and compare it to the labeled percentage. In the plant food, the expected phosphorus content based on the label was 15%. This aligns with the manufacturer's specifications and reflects the formulation of the plant food. But the experiment determined value of 17%, which is slightly higher, suggesting either the presence of additional phosphorus-containing compounds or procedural errors such as measurement error in weighing the sample, filter paper, or precipitate, as well as inaccuracies in volumetric measurements could lead to overestimation. Despite the percentage error, which is 13.33%, the result is within a reasonable range, confirming the presence of phosphorus in the sample. This experiment successfully determined the content percent of phosphorus in plant food through gravimetric analysis, which is 17% with a percentage of error of 13.33% using plant food from Bayer Special Green. The percentage of phosphorus on the label stated 15%, which is lower than the result of the experiment.